focus on troubleshooting and sort of discussing the challenges that we face in our projects thus far. So we have a variety of experts attending today, which have expertise in algae, plants, and cloning and other laboratory methods, and then for all of us to sort of discuss our challenges and advance in our projects.
Okay, so if everything's fine, we can continue with this. So the section now is that we're going to start. We brought two really cool people to talk a little bit more about their experiences. And one of the topics we wanted to talk a bit more today was about overcoming experimental roadblocks. So that's something that's going to be on the background. So if you want, you can go again. I do have to go after this because I had to move out. That's a picture of me trying to move all my plants from my place yesterday. So yes, saying that in advance, I'm sorry. But without further ado, let's continue. And I would like you to present first one of our first speakers. In this case, it's Dr. Luis Stefano. He's a faculty member in UPCH and in Peru. He's a plant biotechnologist. He has a PhD in biochemistry from the Louisiana State University. He is a really big plant GMO advocate, and he's someone that I've been really inspired for a long time, and someone that I admire quite a lot. So without further ado, bienvenido, Luis.
Thank you. Okay, shall I start or?
Yeah.
Oh, okay. Well, talking about how to get around roadblocks in the lab. I remember when I was graduate student, my mentor, Dr. Jesse James, had a very special way of teaching or mentoring. He would always ask me, what do you think? You know, about something that I wanted to know, but he first would ask me, what do you think? And I will go, well, I think I can do this, I can do that. And then he would say, okay, go on. So basically I was on my own, you know, and I think he did that on purpose because he wanted me to be very independent quite soon. But you know, back in the 80s, and especially for me, since I was coming from a country where recombinant DNA was quite new, everything I knew was just from the books. Doing plasmid manipulations and transformation, E. coli transformation or electroporation, you know, was quite difficult, I guess. However, I learned how to get around this. And one thing was to get all the information, get the original papers where they will describe the technology or the technique, trying to read as much as I could. That meant going to the library many times. You know, back in those days, you didn't have Google or anything like that. So you had to go to the library. And fortunately, the LSU library was very good. It had like 5 million books and journals and everything. And you were free to just go by yourself and, you know, look through the shelves. So it was very, very nice. And after reading everything about that technique, I will go around and ask for further questions from the postdocs in the building. You know, those that were already one step further in their careers, and that helped me a lot. Then once I probably master all the cloning techniques and the vector constructions, you know, I'm talking about the days when you did everything using restriction enzymes, no PCR. Okay, this is before PCR. So you have to work with triple ligations, for example, and hope for the best. And I mean, it was quite an experience. And your cassette, you wanted to be in a special direction and not in the other direction. And then you would screen for 20 clones, and all of the clones were in just one direction. Why? We don't know. E. coli didn't like the other direction, you know. I'm talking about in relation to the kanamycin marker. Sometimes you want to have the orientation of your cassette in a special direction. So, you know, so you have to do a lot of work. And as I mentioned at the beginning, getting a PhD is not of being smart or not. I think it's a lot of willpower, you know, and stamina, and some, you know, something that you put all your efforts directed to that, to your goal. After that, when I obtained my PhD, I went to Ghent, to the lab of Professor Van Montague, and I went to work in the ethylene group. And there, I had to do a lot of library screening, something that I didn't do, I haven't done for my PhD. And again, I had to go and look through literature and talk to the postdocs. Fortunately, Professor Van Montague's lab had 170 persons. So there were a lot of people to talk about. Yeah, believe it or not, 176 people working for him. You know, we were talking about the European way, you know, when you have head professors and you have group leaders. So no assistant professors, group leaders. And, but the corresponding author, you know, it was always him. You know? The group leader was the last, and he was the one before the last. And well, anyway, so there were a lot of people to talk and to troubleshoot problems. And another good thing is that you can have all the restriction enzymes you wanted. Everything was just there. You will need something, you will get it in the afternoon, not the next day, but in the afternoon. You will get P32 or whatever. Sequencing, you didn't have to do any sequencing. There was a group that you will just go and, you know, hand in your lambda clone or your sub clones and say, please, you know, I need this sequence, and you will get your 20 kb or insert in a month back in those days. I'm talking about 91, 93. Capillary sequencing was in those days. So, and then other problems that I had when I was a graduate student was plant transformation. You will go and read the protocol, and it looks so easy, you know, cut your tobacco leaves and incubate them for 2 minutes with a overnight agrobacterium culture, and shake it for 20 seconds and blah, blah, blah. But it was not always so easy, you know, and especially with the vectors that we used to have before the binary vectors, we used to have co-integrate vectors. And, you know, just doing the construct was very hard. You have to screen for a lot of recombinants until you get what you really wanted. But when the binary vectors came along, everything looked a little bit easier. And well, tobacco, it turned out to be very easy to transform after a while. And then next thing was potato transformation, a little bit more difficult and cultivar dependent. You know, it depends which cultivar. Desiree, for example, was very easy to transform, but roasted bourbon was extremely difficult. You will try 1,000 explants with no transformants, 0 transformants. With Desiree, it will work like almost like tobacco. So later I went to work in SEAT, and that was almost like a permanent job. So there I had to learn AFLP technique. I don't know if you remember or you know AFLP. Maybe those names tell a lot about my age, because I think AFLP is no longer done. Now you can go around with RNA-seq. And but anyway, so although the techniques have changed, I think basic rules is that you have to read before everything that is about the technique. You know, go to the original paper, read it, try to go and look for the next paper from that group. You know, sometimes they tell you in the next paper some problems that they had before, how they solve it. And then ask for help. You know, sometimes your PI could help you, sometimes not. They are very busy trying to get tenure. Writing papers, grants take a lot of effort. And so postdocs are very helpful. They already gone through the learning process, and they can give you hints that are not published, you know, and they help you a lot. And about about your attitude, I think you have to accept that there's a learning process in any technique. Don't feel discouraged. Try to go to work every day with a nice face, with a nice attitude. You know, just focus on your work, and everything will come into place after a while. You know, and there's always a chance that you will find out a shortcut, you know, and another way that nobody thought about, and it will be a good, nice. It could mean a nice short communication. You know? So I don't know if you have any other questions. I will be happy to help you. You know, Gustavo asked me last night to give you a little bit to talk about my experience, and so that's what I've done. So that's it.
Thank you very much.
Dr. Di Stefano, I would love to know about your favorite research project you've ever worked on. and some of the experimental roadblocks you had with that.
My favorite project?
Yes.
Okay, well, I would say that the work that I did in the USDA, I elucidate the whole ABA biosynthesis. I characterize all the ABA biosynthesis enzymes in potato and characterize the enzymes involved in the metabolism of ABA. I found out that there were 4 P450 enzymes, that will degrade ABA after they do their work, and found out very interesting things like alternative splicing, stuff like that, that eventually I couldn't get further there because in USDA you have the rule that you go for what is important, not for what is interesting. Okay, that's a difference. Because in university, when you are going through for your PhD or you are doing a postdoc, whatever looks interesting, sometimes your PI will tell you, okay, go ahead, and see what happens. But in industry and in a lab, in a government lab, is different. You have to work in your project. You have to do what you've been hired for. And after that, if you have time, you will go for whatever looks interesting. That project was very nice. Our group was very, I never had done before cloning and expression of genes in yeast, for example. And I expressed several of these genes in yeast and isolate the microsomal fraction and do a lot of assays, and that worked very nice. That worked in the first attempt. It just everything went boom, 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 boom. And from that project, I think we published like 3 papers. So that was
very awesome. Well, thank you so much for that talk. Saged words of wisdom. It's nice to know that, you even back then you had the same struggles as we do sometimes.
Well, I'm from the generation that we coined a word that I think is no longer used in academia or in research. They would call him gene jockeys. So people will go and chase for a gene, characterize it, clone it, and try to do everything about one gene. Now you can do many genes at the same time, But one last thing that I want you to know or to internalize is that in biology or in science, it doesn't matter how good expertise you have with your hands. What really matters, okay, especially if you are going to have your own lab, is the kind of questions you are able to come up. Okay, that's what put you, apart from the group, from the pack. What kind of question you are able to ask about a problem. What if? What's important about? And many people made the mistake of learning a technique and they fell in love with that technique, okay, or with a group of techniques, and then they look for a problem. Okay, so they are experts in a technique and they say, okay, now I have these weapons and now I want to go to war. No, it doesn't work like that. First, you have to find out what kind of biological process you fall in love or you are really interested. And then you look for the weapons you need to go into battle, you know, and then you will go and choose wisely. Okay, maybe you don't need something really fancy. Maybe you need something old-fashioned like a gene screening, you know, or something very basic. Okay, and then ask questions from there. Okay, so I remember that what matters is what kind of questions you are able to ask about a research topic. Okay, forget about the technique. Forget it about it. Okay, later you can choose the techniques or the experimental approach that you want to use.
Awesome. That was awesome to hear about. Thank you so much for sharing that. Just a quick introduction of myself. Hi, I'm Robin. I am a member of the UBCO iGEM team. I mostly work in wet lab with our phototrophs. And next up, I would like to introduce Dr. Mitra Tabatabaee. Dr. Mitra is a sessional lecturer at UBCO specifically, our campus. She specializes in microbiology of built systems, like signaling chemotaxis, specifically chemotaxis in brain cancer cells. And she has a PhD in both microbiology and molecular neurobiology. She is one of the PIs for our UBCO iGEM team, and someone who I work with a lot personally, actually. And she will be talking more generally about overcoming those experimental roadblocks and as that relates to her PhD experience. So Dr. Mitra, take it away.
Thank you very much. May I share my screen?
Oh, please go ahead.
Okay. Okay, so let me just perfect. And let me do something. Okay, thank you very much for introduction. And congratulations to you guys that gather together on weekends to talk about science. It is amazing. This is the first step that you're taking, and this is one of the, I think, the most important step that you're taking. Thank you to Robin for the introduction. As she said that I've done all my life, I've spent all my life doing PhDs. So after doing my first PhD in microbiology, I did my first PhD in Middle East. And it was specifically an industrial microbiology. And then because of this question that I had, I can talk about it later if you are interested to know, I decided to go for molecular neurobiology because I was finding some common features among the bacterial life and the specific type of cell in the brain, protein cell in the brain, that I was thinking that it plays role in a cancer, that it did. But for doing that research, I had to have knowledge in neuroscience. So I did my second PhD. And right now I've just completed the second one. After doing the first one, I worked as an assistant professor for a couple of years. And then I returned back to school again as a student. And now I've just started at UBC again as a prof. I told you I'm not very much ready for this talk, but I have, you know, that I always have a lot of story to tell you. So I tell you my stories. But before going to the stories, I want to tell you one very important point. When we are talking about science and becoming scientists, is the difference between a chef and cook. Do you want to be the cook or do you want to be a chef? Nice. To be a cook, you just need recipes. You need a book, cooking book, open it, follow the recipes, part 1, A, B, C, D, and then add this much of this to that much of that. And at the end, the final product is either good or bad. Usually good because it has been troubleshooted by someone else and had tried it before and optimized the technique. So they have written the book and gave it to you. But when you start doing the science, you should ask yourself, do you want to stay a cook or do you want to be a chef? The difference between chef and a cook is that when the chef comes and look at your product, I mean, your food, your meal, the plate that you have prepared, just by smelling that, he or she can say, okay, the turmeric is not enough here. Or when you were mixing salt, you had to add the pepper right away there. Or if you add salt first to your oil, it avoids, for example, sticking. Some tiny points that you can never see or find them in the protocols.
Mhm.
So this is the point. First, you have to decide whether you want to be a chef or a cook. And the difference between being a chef and a cook make you an industrial scientist or an academic scientist. And industrial scientist, as Dr. Stefano pointed out, is just knowing the techniques. You know one technique perfectly or a couple of techniques, and you use them daily. On a daily manner, you just practice what you know and you get better and better and you're being paid more and more. And one thing is that because you are expert in that field, you're being paid more. And you know that usually industrial scientists are much more richer than the academic scientists. And this comes from your interest as well. When you're an academic scientist, you want to become a chef, regardless to the payment. You want to quench your internal thirst for knowing, for answering the questions. So what is important for becoming an academic scientist, and what I always want you to do, is first asking a question. Do you remember the members of UVCO, iGEM? We always try to start our meetings with questions. So you should be mastered in asking the questions, the right questions, and variety of questions, and rewarding your questions. It means that sometimes you ask one question, but you can't answer it. So you should find a way to go around and reward it. Use some other words, look at their question from different aspects. And this is the art of an academic scientist. So first of all, you need to ask the right question. Why I am doing that, how I am doing that, and what I'm expecting to reach. The bigger the answer for why is, the more, that driving force would be stronger. It means that, for example, the why question is that, okay, I want to solve a global problem. It is a big goal. You may not be able to do that. You may just be able to, I don't know, manipulate one single gene or transform one single cell, or as what Robin's doing, trying to draw the growing curve. It looks small, but when the goal is big, you keep going. You do your first PhD and you do your second PhD and you may go more. It is that the question is that big. Probably you cannot reach that point. Probably the point is that you have to do your best to answer your own question. The whole story the point here, I said that the mindset. The whole story of being a good scientist is your mindset. Why are you there? Because it's a prestigious work, prestigious job, good paying, and then lots of respect. Or this is because you are a good student, you learn things well, you are an analyst, you have an analytic mind. Or this is because you want to do something. This is because you want to make a change. So you have to think, all of them are good. I don't mean that one is good, the other is not bad. Even if you want to work for a better payment, it means that you're thinking for a better life for you, yourself, and the people around you. All these goals are very respected goals, but you should define it for yourself. You are right now in iGEM. You're working, I can see that you're working days and nights. You start your days at 6 a.m., end it up at 10 a.m. or sometimes receive some messages from you at 10 p.m. I said, okay, amazing. You are on the right track. I know that some of you do not sleep overnight just because you want to keep yourselves alive. And this is the right track. But all the things that you're doing should have a big goal. So you should think for yourself, okay, why I am here at iGEM, why I am spending this time, why with these people, what I can learn from the people around me rather than science. Science is important. Definitely science is important. But more important than science is your mindset, because you need the driving force. It is so hard. When the first block that you hit stops you. No, it doesn't stop you. It makes you tired because you have to push. The second one, you ask yourself, what I'm doing with my life? Why I'm here? Lots of people are working out there much easier and earning more. Why I'm doing? And the third one that you hit, you stop. The only thing that can keep you going, push you going, is the mindset to have. It is not a part of troubleshooting. Yes, it is a part of troubleshooting, because then it shows that how much energy you will put to troubleshoot when you face a problem. The second thing is knowledge. Knowledge doesn't need IQ over 200. I have always told you, and I always tell all my students, if I have a PhD, doesn't mean that I am smarter than you. It means that I am older than you. I have spent more time on that. It means that knowledge is something that you can simply acquire. The time that you spend watching Netflix, if you spend reading a book that you are interested in, you become knowledgeable. And I say interested in, I literally mean that you're interested in. You can spend time reading a novel. That's great, from great science novelists. But sometimes you enjoy reading books, for example, in neuroscience, enjoy reading books about photosynthetic bacteria. It is your joy. When it is your joy, that knowledge becomes a part of you, and then you don't, it's not suffering anymore. And when I say knowledge, I mean, first, a broad knowledge. Probably for me, it is easier to say that because I have switched from one very different field to another very different field. I was working on petroleum microbiology and then switched to neuroscience. It looks very different. But when you go at the molecular level, there are that much common features in these two sciences that makes it an amazing nature that you can't stop learning about them. You're always thirsty to learn more about it. But the knowledge, what knowledge does for you, it paves the way. It makes things much, much, much more easier for you. It is just like, I just remember my son when he started learning how to tie his shoelaces. He was struggling with it. Every morning he had lots of cries, tears, and fears of, I can't do it. But then just by telling him that, okay, what we're doing with tying is we are trying to put this part of lace behind the other part so they don't get apart. So when he understood the basic and the fundamental of that, just by 3, 4, 5 trials, he learned it. And then even if it didn't become his joy, but his second nature, he doesn't think about it, he just ties it. This is the same thing about science and doing scientific experiments. If you know the fundamental, the foundation of what you're doing. So what I'm thinking, I'll talk about it later again, but about the knowledge. When it comes to troubleshooting, knowledge is the only thing that helps you. And knowledge not about the techniques, knowledge about the fundamentals. For example, you want to transform E. coli, and it doesn't work. If you know the basics of transformation, that the plasmid should be attached to the surface of the cell should have all those superficial components on the surface to take the plasmid. I mean, if it has some pelees or something that can take plasmid or sticky compounds on the surface, it makes it easier. If not, I have to help the cell to take it, to attach and then take it in. So when it comes to taking the plasmid in, is the cell ready, or is it porous enough to take the plasmid in, or I have to reduce the surface tension of the cell to help it to take in the plasmid. I mean that if you know the process, you can easily troubleshoot it. Not easily, when I say you can think about all the different components that is making that trouble for you. And you should be creative. This is what I always encourage with students. Usually the scientists who have a little bit artistic talent are more successful scientists, because creativity is a very, very, very important part of solving the problems. You don't always have access to everything in your lab. You should think about the things that you have, your availabilities, your resources, and think about the problem, and then find a way to use the resources that you have to solve the problem. Talking about that, I remember my problems, the issues that I had when I was doing my first PhD in Middle East, and we really had lack of resources. We always have to think about the way. I was trying to isolate methanogens from deep subsurface, 10 kilometers below surface, and they are extremely anaerobic and very sensitive, I can say, squamous cells, that you have to pay lots of attention to them. So we were taking them up, but right away in exposure to the pressure outside and the air outside, we didn't have anything. We did the same thing for months and months, and we didn't come up to anything. So we had to think about the way. And yes, it was some technique at those days, some machines that could go down, drill, and take in 100 tubes the samples, but we didn't have them. So we should think about very single ways. Use the hoses and some simple vials, cream seal them. I mean, find a way. Or I remember that the first time that I designed my anaerobic digester myself in the lab, and I asked my supervisor, I showed her, I showed her, I want this. And she said that, okay, this is not our job. We need some artists, some glassworks. We need someone to create it. And then we asked some people who were working with glasses to create that, I can say, construct. It was made of an Erlenmeyer, some glass cords and wirings that could connect these different parts. What I mean is that if you want to reach the answer that you're looking for, you should use your creativity. Now, I'm just telling about my experience. This is the same case about your experience. Although I know that you're creative enough, specifically when all of you have designed some, a thought about some projects that are good enough to compete with the others, it means that you're creative enough. However, try to work on your creativity and passion. The thing that I told you that you should, this is the mindset. This is the part. That's why I'm doing that and what I will reach if I can do it. Even if you can't reach it, you don't reach it and you won't. But this idea that if I can do it, what a big movement it is. This is the passion that you will have. And patience. Patience. Years and years. Sometimes Dr. Stefano knows how much time sometimes you have to spend to do one single experiment and not getting the answer, and then you have to troubleshoot and do it over and over and over and over. So patience is the other part, and bias for action. You should act. Just with sitting, thinking, asking questions, and reading is not enough. You should also act. So I talked about the differences, but now this is the where you have reached. You have the question, you have the path, you know what you want to do, and you have chosen the type of experiments that you want to run and you need to do. You either have the resources to do that or not. If not, then creativity comes to play. You have to think about other ways to answer the same question, look at the same question from different angles. But just suppose that you have the right question, you have the right resources, you have started your experiments and you ran it, and it doesn't work. And it doesn't work. And again, it doesn't work. Try. So first try it more than once, because sometimes these are very simple, crazy reasons that problems happen, but and just by repeating that, it will be resolved. But sometimes this is more complicated. But before repeating that, you have to start thinking about the possibility, because it's possible that the second time it doesn't work. So first you have to identify. There are some steps that you have to do. Identify the problem, find it, try and eliminate all the irrelevant possible problems, and then fix it. What do I mean? Identify, I mean, look at the symptom. For example, you have done everything right as you think. I talk about transformation again, the simplest one, and then your cells don't grow. Lots of questions. It doesn't grow. It means I can't see the final, it's the worst part actually, the final product. It means that from the very beginning, something might have happened. These things could be the problem with your protocol. Maybe the protocol that you're using is not the one that perfectly works for you. I mean, just by changing the bacteria, maybe it doesn't work anymore. Maybe just by changing a couple of degrees of the temperature, maybe the protocol doesn't work. Maybe even some very simple things. Maybe the Eppendorf that you're using is not clean enough. It has some impurities in it. These are very simple things that you can think about the protocols that you're using. Just I can give you an example of myself. A couple of days ago, one of the members of Dr. Marin's lab came to me and he asked me that you have published a paper and this was the protocol that you did the calcium imaging. And I tried it over and over. I did everything right, just you, but it doesn't work. And the point was that when I was adding DMSO, instead of shaking that, I just inverting the Eppendorfs to mix it. And it was not a thing that I was published or written that in my paper and published it. And no one else could tell him except me. And it was a thing that I didn't even not think it's an important thing, because what I was doing was working. It was perfectly working. And when he asked me, I was thinking, yes, it is important. Just because DMSO can kill the cells. And when I shake it, I start making the cells porous and I lose all the cells right away before doing any experiment. But I just want to mix it in the media. So just few inverting was enough. What I mean by identifying is that, I meant that it is true that you have the protocol, but the very tiny, the ones that are not tangible points that play a role in your final result. And you can tell all these based on your the symptoms that you have in your experiment. Then you have to start to eliminate non-issues. For example, check if your other compounds that you're using are okay. They're not expired. They are all work perfectly. All the glasswares and plates and whatever that you're using is clean enough for what you're doing. When I say clean enough, it's not just washing. Sometimes you need to acid wash them. Sometimes you need to use acetone to wash them. It means that what you're doing with your dishes that you're using. Sometimes the apparatus that you're using is not calibrated and there is something wrong there. So you first have to eliminate non-issues and then return back to the protocol. And then protocol, you can then try to optimize the protocols and have that in mind. All the optimized protocols for others may not necessarily work for you. So you need something, many times you need to optimize it yourself. And then you have to start to find a cause. And find a cause is that pain in the neck. So it means that you have to go step by step. Let's start with the most obvious one, the ones that is more common. And how you can tell that which issue is more common is just, it comes from that knowledge. The more you had read about it, the better you can tell where the problem should have come from. And then fix it. try the first possibility. If it didn't work, the second one. If it didn't work, third one. I mean, sometimes it is easy, but sometimes it is not that easy. You should be ready for that. This is where you need the mindset, being patient and having the passion. But asking for advice is maybe sometimes the best way. Asking the advice from even the people who are not exactly in your field. When I say you don't exactly need molecular scientists if you face a problem in molecular biology. Maybe a plant scientist, I don't know, a chemist, a biochemist, a cell physiologist, because they are looking at the same object that you're looking at from different angles, they may see and notice something that you have never noticed that. So communicating is another very important thing that can help you. Just I remember something, just I said, no, communicating, I remember that. But I talked about knowledge. Knowledge does not necessarily come from the books or papers. Knowledge can come from the people, can come from the conferences, can sometimes even come from the news. The more you know, the better you can analyze. What else? I have a lot to talk about right now, but I can't remember. How can I stop sharing my screen? No, stop sharing.
Yeah. You may have some option around.
So first, let me stop sharing. I don't know how to stop sharing my screen. Why doesn't it work? It just comes share new, but not stop sharing. Okay. It post share, stop share.
If you
Oh, okay.
There we go.
Good. Yeah.
Cool. Thank you very much.
So, any question? Ready to take your questions.
I guess you were talking about how sometimes when you're following a protocol, it doesn't work because you have small mistakes along the way. And our research group, we're working on transforming our algae, and the protocol isn't really working for us at all, and we think we might have those small mistakes. I was wondering if you can elaborate more on how to identify like those potential points where you're maybe doing something that's a little bit different from the protocol, or something that might be slightly wrong that could be like ruining your whole process.
So you're using an algae, yeah? And the protocol is for algae as well, or the protocol is for something else?
Yeah, the protocol is for algae, and it's for genetic transformation with like glass beads.
Oh, glass beads you're using. Okay. So you should know, for example, tell me, I ask you, you can troubleshoot yourself. How do you use the glass bead method?
So we put a few small glass beads in a glass test tube, and then we vortex the glass beads with our DNA and our algae. And
why do you use beads?
We use the beads to sort of, I guess, create, so when we vortex it, it creates enough pressure to like create like an opening in the algae membrane, and then
what do you mean by make opening? It means that it rips the membrane?
Yeah, a little bit. And then because
that's okay, the first one, it can rip the membrane. Maybe you have shaked it too hard. Okay. Yeah, go ahead. So, and then what do you do with your beads?
We remove the algae from the beads.
No, first you cover your beads with that DNA, yeah?
Yeah, we cover the beads.
How do you do that?
We just pipette the DNA onto the beads.
How do you know that it has attached?
We do not. We just
how do you identify that? Usually, typically, when the people use the same technique, how can they identify that the DNA has been attached to the beads? Go on, find the answer.
Oh.
Yeah, it is very important. Probably you don't have any DNA there. You have put them together, you have mixed them, but it has not been attached. Find a way that whether it has been attached or not, and then if it is attached, the affinity is also important because it has to go through the next vigorous step. So it means that maybe DNA is not DNA attached. You just put the bead, and the bead just hitting the membrane, and DNA is there, and by hitting the membrane, it makes it porous so the DNA can go in. It's not necessarily always need to be bound on the surface. So now it is two options. Probably it has not been bound, and the other one is that now you want to hit the membrane. What technique do you use for that?
We're not heating the membrane necessarily, but I guess like as we vortex, maybe
Vortex.
something could
So
happen.
how vigorous do you vortex it?
We vortex it at the maximum speed on our vortexing.
How do you know that maximum speed and those many beads do not kill yourself?
We tried to do a transformation with no DNA and no selection marker and just plating our cells after, and they did survive after, so that kind of told us that just the vortexing process itself wasn't killing it.
Perfect. So it means that you know that the vortexing process, it is eliminating. You are eliminating the non-issues. Okay, vortexing is not problem. And the problem is that the DNA cannot get into that. So how big do you use the DNA? Is in the format of the plasmid, yes?
Yeah.
Yeah. How can you tell that your plasmid is not broken during the vortex?
So I guess I don't know how to tell if the plasmid doesn't get broken, but I guess like if the plasmid does not get into the algae, or if it does and perhaps it gets damaged, then the algae would not have resistance.
Okay, if I were you, I was thinking about one of the troubles. Actually, I go over all. I start from the simplest, and this is one of the hardest that I'm saying. For example, here I think that, okay, it is possible that the plasmid is not in the right format, and the plasmid is broken, and this is why we don't see what we expect to see. So what I do after vortexing, I would run a Western blotting to see, or a blotting, or DNA blotting, I mean, any type of blotting. You can run electrophoresis to see that, do you see that length of the DNA that you expect to be there even after vortexing? If not, the problem has happened before. So the point is that what I'm saying is that if you know the protocol step by step, then you can think about the possible events, what had happened there. What about yours? I don't know because I have never used this method. I usually use the heat shot technique or electroporation. The reason that I don't like beads usually, because I told you I have used the brain cells, and the brain cells are very sensitive. When I say brain cells, I have to use the E. coli, and then I transfect the eukaryotic cells. I mean the brain cells with the plasmids that are coming from the prokaryotes. So the brain cells are very sensitive. Everything, even I told you flicking. Moving your hand, every single action of you can kill them. So it is important to consider all of them. I have never used those beads because it can simply kill the cells. But you can troubleshoot by just thinking about the steps and whys. Why in this protocol this specific step is being used, and then think about the possibilities. And talk in the group. And group working is very important. For example, the thing that Dr. Stefano mentioned about a leader and the group, the beauty of working in the group is that you don't have a pair of eyes anymore. You will have 100 pairs of eyes, and each one of them can have a different idea. Sorry. Okay. Any other question?
Thank you so much. You gave us a lot of good ideas about how we shoot.
Dr. Mitra,
Yeah.
I would love to ask you a question. I have noticed that the questions that you ask yourself, teach us to ask ourselves, is some of the questions you've asked me. Mm-hmm. Now, if you were me, how would you ask you, or I guess in a more broader sense, how would you ask an expert a question in general?
Oh, very good question. but not easy one. How to ask from the expert. So this is exactly, you can use the exact thing that Gabriela did. You can ask, you bring up the problem, talk about the issue. This is the finding part. You find it, the symptom. Yes, the point is the symptom. You can say, okay, this is the symptom. And then you should always, when you talk to the experts, you should have something in hand. This was the symptom. I tried this, but I have not found any answer. This helps that expert to think about the things that you have not thought about. Otherwise, you will kept in a loop. You have thought about some troubleshooting techniques or some possible ways to get out of that situation. But that person that you're asking doesn't know that how you're thinking. But when you have tried a couple of them and you ask the expert, then the expert can, the first thing that happens is that they can evaluate your knowledge. They can know, okay, how much you know. And when they know how much you know, then you can give you, even if they don't know the answer, they can give you the direction. I'm sure about that because I told you I have done 2 PhDs and I had lots of supervisors, co-supervisors, committee members, and this is what I have learned from them. Sometimes they are not expert in that field at all, but when I go there, tell them the problem and tell what I have done about it, then they help me. Just one other thing that I remember about my own experiment when it comes to troubleshooting. One problem that I had, I was working with an astrocytes, a type of brain cells, that I couldn't keep them alive for long. And the process that I was doing, I wanted to try one specific drug on them to see their morphological behavior. So they had to be happy and healthy at least for 45 minutes that I was filming them. And it was not happening. After 30 minutes, these cells are sort of, I cannot say immune cells, but they are also active in immunity. So they change their shape with lots of processes and they shrink their body. And I couldn't see their reaction anymore. Then I realized that the problem was with the heating stage. I tried everything, but I thought, okay, the temperature was dropping in time. And they are the cells that should stay at 37 degrees. So what happened, I went to the mechanical engineering department. I told them, this is the problem, help me. And what they did, they made homemade heating stage for me. I still have that. This is very funny. You can see that how they have cut it with in a piece of steel. And then we had lots of problems because they fit on the surface of the microscope, and then the thickness of that was also important because I was using invert microscope and I wanted to the microscope lenses reach the bottom of the glass bottom plates. I mean that the point is that you don't necessarily need to ask when you say experts, the person who are in that specific field. But when you ask with the people who are in that specific field, they can give you the clue whom you can go to and who you can ask help for.
Awesome.
Okay, anymore?
Very informative. I would love to open that question up to Dr. Stefano as well. Dr. Stefano, how would you ask a question to an expert? Like if you were me and I was asking you a question, how would you best approach that?
Well, I will try to first well, depends, if I meet this expert in a seminar, first I will find out what this person is expert in what field this person is expert. And I will just go over their research interests and what he or she is doing for the last, let's say, 4 or 5 years. And then during this, after their her or his seminar, I will try to ask a very smart question. One thing that is important in graduate school is trying to attract the attention of your faculty, of your professors, or your postdocs, your your possible postdoc's boss. If you want to do a postdoc in a specific lab, usually you meet that person during the seminar labs, on Fridays or I don't know, Mondays or whatever. And you want to prepare yourself and do a very smart question, something that is more than just a technique or something very specific. It's something that will go into the field itself. A more no philosophical question, but a deep question.
Awesome.
Something that will give the person that you are asking the question a sense that you have some gravitas, okay? That you have some bone in there, to be a good scientist. Never try to ask a question, something very simple, oh, did you run these controls or did you try this other thing? Or no, try to go into a more deep question. I always did that when I was in graduate school, in those 8 o'clock in the afternoon. That was the time when we have these guests every week coming from all over the world.
Awesome. Thank you so much for that advice. I think I'll definitely be making use of that. So for everyone who's just joining us, I think we're going to get into our troubleshooting breakout sessions now. So basically how we're going to structure this is that we have 3 experts for you guys to take advantage of, and we're going to divide them into breakout rooms. And, teams can jump between breakout rooms or join them as they see fit. And one of our experts is Dr. Luis Di Stefano, Dr. Di Stefano, who's our plant expert. And we also have 2 other faculty experts for you guys.
Dr. Everett Eustance is an assistant research scientist at Arizona State University, and he's the faculty associate at ASU's Polytechnic campus. And he's been researching at ASCADI, which is a research center focused on algae, for almost 15 years. And today he'll be helping out with the breakout rooms focused on providing help for algae. We also have Santiago Ochoa, who is a PhD student in biomedical engineering at ASU, and he's in the lab of Dr. Bartel. And for those of you who don't know, he also participated in iGEM as an undergraduate, so he has experience with this. And today he'll be helping us in the breakout room focused on cloning. So we have opened all the breakout rooms for you guys, and you can join whichever breakout room your team needs help on. I'll share my screen again so you know which one corresponds with which. So breakout room 1 is for algae, room 2 is for plants, and breakout room 3 is for cloning. So join whichever one your team needs most help with, and feel free to switch between them.
Okay, I will join room number 2, and I want to say goodbye and to all the people in the group. Okay, so has been very nice talking to all of you. Okay, have a nice day and happy weekend.
Nice to
Yes,
meet
thank
Bye-bye.
you so much.
So much, yeah.
Bye.
So now, like I said, we're going to wrap up by talking a little bit about the handbook and video contributions. So recently we sent out the form where you can submit your proposals for either your video or handbook contribution to the 2022 iGEM Phototroph edition of the handbook. We sent you all back proposal feedback regarding your submission form. Either we said you're okay and we think you can go ahead and start producing either your video or written contribution or both. So if you got that okay, you're good to go begin that process and your team can start working on that. If we had some clarifying questions for you, just be sure to get back to us regarding that and finalize your proposals by August 23rd. And then after that, once you've had your proposal finalized with us and approved, you can go ahead and work on that whenever that gets done for you. September 1st, we're gonna have a progress check-in just to see how things are going with you and see if you need any like clarifying updates regarding the criteria for the video and protocol additions. And then September 25th is when we'll have you turn in your final productions to UBCO and ASU, and we'll send in further contact details and submission details later on about that. And then on October 5th, that's when we're going to have the handbook complete, and we're going to send out badges for you to display on your wikis, and we'll have further information regarding that. But just before I move on, does anybody have any questions about this broad timeline? If not, I'll move on. So for those of you who are doing a video contribution to the handbook, we do have a few criteria and requirements for your videos that I wanted to reiterate to you. So your video must be 2 to 10 minutes in length, and it must be filmed in landscape. And we strongly recommend voiceover over just filming your audio as you're taking your shots. Because the quality of the audio will be a little bit better if you do a voiceover, if that's accessible for you. And please contact us if you have any questions about that specifically. And we would like all of your videos to start out with a provided introduction clip that we'll give to you, kind of like for your iGEM promotional videos, how they provide you with an introductory little video to put at the beginning of your slides. So a lot of these requirements are slightly similar to those for the iGEM promotional video, but there are some differences, so please keep this in mind. But if during your video production process you have any questions about any of these requirements, or if there's something we maybe missed, then please definitely let us know and we'll get in touch with you and clarify that for you. But otherwise, are there any questions about the video contribution requirement? Okay. If not, thank you so much for attending this second meetup. We hope you got a lot of your questions answered for your teams by talking to our experts, and we hope that our general speakers inspired you to continue troubleshooting your projects, even though right now you might be facing a tough patch experimentally. But we hope that this provided you a lot of insight and a lot of motivation to keep going with your projects until the Jamboree. So after that, we just wanted to thank you and remind you again to stay active in the iGEM Phototroph Community Slack, and if you haven't already, join it, and also to keep your eyes out for information regarding the handbook submissions and also the final conference, which is coming up next Saturday. Thank you so much.
Awesome. Yeah, thank you guys for attending, and thank you to ASU for co-hosting here. Yeah, I think that just about wraps it up. I hope everyone has a delightful day.